Okay, so the title of my presentation is The Identification of Necessary DNA Promoter Elements in the Thymine Biosynthesis Pathway of Candida glabrata. It's the wrong title. So why study Candida glabrata? Candida glabrata is a pathogenic yeast species, and it's an opportunistic pathogen, which is an issue in hospitals for people with suppressed immune responses. And it can cause candidiasis, which can cause thrush, like this disgusting picture over here. And it also has a high drug resistance. So there are drugs that target other yeast species in the Candida family, but not necessarily this one. And it is related to Saccharomyces cerevisiae, which is a really well-studied yeast species as well. And it also has a very interesting thymine pathway, which is what is my project will be focusing on. So what is thymine? It's a vitamin B1, and it's essential for life. It is produced by yeast, but it's not produced by humans, which makes it a good target for antifungals. And it is active in, wow, that's the active compound. And it is necessary for amino acid synthesis and metabolism. So this is a thymine biosynthesis pathway in Saccharomyces cerevisiae. It's a complete pathway. Cerevisiae makes thymine really well. Glabrata is missing some of these genes. It's still functional, and it can make thymine. It's just not as functional. So previous study in our lab was done on the genes in that particular pathway to see which ones are induced in thymine starvation conditions. So they were grown overnight in thymine starved conditions, and then RNA-seq was run to see which genes were upregulated, and the genes that were upregulated are the genes that are active in that pathway and are the ones that are induced in thymine starvation. So those are the genes that I will be focusing on. So there's two different transcription factor regulations in glabrata and cerevisiae. So essentially, what I'm looking at is the location that these two bind in. Since I'm looking at glabrata, it's Thy3 and PDC2. And in order for these to be active, the transcription factor has to bind them, as you see here. So when thymine isn't present, these two transcription factors will bind to this location in the promoter that I'm looking at in order to cause transcription and turn on the genes. Oh, I don't need this slide anymore. So my project is that there is a binding site in the promoters of all of these genes that is essential for upregulation, and then I need to change this one. But I'm predicting that these locations are similar throughout several different genes, and that they can be mutated to eliminate upregulation. So how am I going to do this? So essentially what I've done is I've created plasmids to amplify the desired sequence. I've prepared these plasmids and transformed them into yeast, and then I've starved them and used flow cytometry to determine the fluorescence. So further, to go into more detail, essentially what I've done is I've taken a specific primer that is in a location that will truncate the promoter to a certain length, and then I have another primer with similarity to YFP, and then I make this section of a truncated promoter, and then I put it into a digestive plasmid that will then be put together as one in yeast with plus His so that you can keep the plasmid in the cells. Once they're transformed into Candida glabrata cells, wow, they'll be grown in starvation conditions of plus and minus Thy. Plus Thy is the control, minus Thy is what I'm actually looking at. And then I will do flow cytometry. So in flow cytometry, a single cell is brought up into the machine, a laser is passed through it, and then it measures fluorescence for 10,000 events. So essentially what you're looking at is that if it's on this side, if the graph is more towards the front, that there's not a lot of fluorescence, and if it's towards the back, there's greater. If it's bigger. So how I'm looking at this is that, as I mentioned, each of these primers will truncate this promoter by a specific amount, and then I'm looking for where there's no longer fluorescence, and you can see that more clearly in the results. So this is PET18, and it's clear that this is where the promoter sets off between 320 and 310, 330. So then I truncated that further to see if I could fully narrow it down, which is done down here. And then I took these locations, so this is another one in Thy10, and then this is the third one in Thy20. And so then I took these locations, and then I deleted them out of PHY-10 and PET18 to determine if they were truly necessary, and clearly they are, because once they're deleted, there's no expression at all. And then I took those sequences, those exact locations, and compared them. And as you can see, there's a consensus sequence, which is really exciting, which means that there actually is some consensus sequences. Yay! So then, this is the sequence through all of them that's matching. The highlighted part is matching this conserved sequence that's true throughout. So the idea is that we could take, don't need that slide, can take this sequence, I can take this sequence and put it into another gene and make it thymine regulated. So that is something that's going to be done in the future. It's already something that's kind of going on now, but that way we can understand that these locations are actually regulated completely by thymine and not by anything else. So other future work is going to be to actually sequence the plasmids that I use to delete, and to delete the plasmid or delete the sequence from PHY-20 as well. And then to look at more genes in the pathway and see if this conserved sequence is true throughout. And then also to study the transcription factors, which is already an ongoing process within our lab. Okay. Yeah. So these are my developments. Wait, I have a question slide.
Stop recording.
Yeah, that was only 6 minutes.